Hello, everyone. We're going to be reviewing proteins, and then we'll move on to transcription and translation. First thing I need you to do is get out your notebook. Name 2 proteins we learned during our small groups. I believe we did this on Thursday, not on Tuesday. Go ahead and type those in the box on the right. The reason we need to know about proteins and examples of proteins is because on our next test, you're going to have to list some examples. So I wanted to make sure you had those in your notebook. All right, next up, we need to label each picture as DNA or RNA. So on the left-hand side, that red molecule, is that DNA or RNA? Good, that's RNA. So that means our orange molecule on the right here is going to be DNA. Awesome. Which base or which letter is not found in RNA? So RNA has all the following letters except what? Good, RNA does not have a T. Beautiful. Which has ribose as its sugar? Is that RNA, DNA, or both? This would be RNA. And then which can leave the nucleus? RNA, DNA, or both? This would be RNA. What are proteins made up of? We see all these little beads, these building blocks. What makes them up? What are the building blocks of proteins? These are called amino acids. And remember, amino acids make up proteins. And then what makes each amino acid unique? You can't circle it here, but just select the right answer. Is it the amino group, the side chain, the R, or the carboxyl group? It's the R. So everything down here is different depending on what amino acid it is. Here are some examples of amino acids. You can see this red R group is different for each one of those amino acids. Where in the cell are proteins made? Which part of the cell? The ribosome. So the ribosome makes proteins. What about folds proteins? Which one folds proteins? This is the ER, the endoplasmic reticulum. Why does each protein have a specific shape? Has a specific shape, so because it has a specific job or a specific function. So the shape determines what it's going to be able to do. All right, today we're going to be talking about how we make proteins. And in order to talk about how we make proteins, we first need to talk about transcription. Transcription is the first step in protein synthesis or making proteins. If you don't already have your notebook out, go ahead and grab it. Title for your notes is going to be Transcription. The title for your notes is Transcription here. So that will be your title. Don't worry, I don't expect you to know what transcription means right now. We're going to talk about it in a second. All right, here's our problem. You are in the kitchen. Your brother's in the basement with the recipe to make chocolate chip cookies. Remember how we talked about cells, right? And how DNA has the instructions to make proteins, kind of like it's like the recipe to make chocolate chip cookies. Well, your brother is in the basement. He's got the chocolate chip cookie recipe. You're up here. Neither of you can move. How do you get the recipe? Any answer is correct here, so go ahead and grab that. Write down an answer in that box. So usually there's a couple different ways. Some people say, well, you could yell. You could call on the phone, text it, make a paper airplane, get somebody else to bring it to them. All those are fabulous answers, okay? The main thing that we need is some way to get the message from here to here. And that's kind of what the cell has to do. Let's take a look. Our instructions, our DNA is in the nucleus. We know that that's the instructions to make proteins. The problem is the proteins are going to be made in the ribosome. So we got to get the recipe, the instructions, to the ribosome. But this ribosome can't move. And the DNA can't move, and the nucleus can't move. So what are we going to do? We're going to send a messenger, right? We're going to send another molecule to send the message to the ribosome. Okay, we send a messenger. Now, what is this molecule called? Well, the messenger it's going to call, or the messenger it's going to use is called messenger RNA. So we said the title for these notes are transcription. Go ahead and write this down. What is this process of transcription? Well, it's when we take the message from DNA and we convert it into messenger RNA. We give it to the messenger so that it can bring it to the ribosome. This whole process of taking the info from DNA and putting it in this messenger molecule is called transcription. So where does transcription happen? I knew this diagram looks super overwhelming. Don't worry about it too much right now. This happens in the nucleus. Where do we make this messenger molecule? In the nucleus. So we see our DNA molecule. We're going to make our messenger. Our messenger is this blue guy. And the messenger is going to go to the ribosome. Again, don't worry too much about this crazy diagram. All we're talking about here is in order to get our DNA message to the ribosome, we have to convert it into messenger RNA first. So how does this work? How do we make this messenger molecule, this blue messenger molecule? It's kind of similar to how DNA copies itself. So again, go ahead and write this down. Here's what we're going to need. First of all, the DNA is going to unzip. So remember, DNA has the code for proteins. It's got all these letters, A's, T's, C's, and G's. And the DNA is going to unzip, kind of like how it does when we have DNA replication, when we need to copy our DNA. After our DNA unzips, you'll see there's this other strand that we're kind of making that attaches to it. So an enzyme, a helper molecule called RNA polymerase, is going to make this messenger molecule. So our DNA is unzipping. How are we going to get the message out? Well, we're going to connect the matching letters, and we're going to make a new molecule that just connects and has the opposite letters of the DNA. So here we have a C. Oh, we're going to put a G because it's still the message. It's just the opposite letters. And it's this RNA polymerase, this enzyme that makes this messenger RNA strand. Now, after it's all done making this strand here, this messenger molecule that's going to go to the ribosome, the DNA is going to zip back up. So it comes back together. That's step 3. And then the messenger molecule, that single strand, is going to leave the nucleus and go to the ribosome. And that's where proteins are going to be made. Remember that our whole goal here is to create a messenger molecule that can bring the message from the DNA into the ribosome. So here, the message is in this messenger RNA molecule. It's just the opposite letters, okay? Anywhere there was a G here, there'd be a C here. Anywhere there's a T here, there'd be an A here. You might notice there's U's over here. That's right, because RNA has U instead of T. So let's take a look at this diagram. This is showing us transcription, okay? We don't see the nucleus, but this would be happening in the nucleus. Take a look at this molecule that I colored blue. Notice it's a double helix shape. It's got 2 strands. What molecule is this going to be? Is this DNA, RNA? This is DNA, right? It's a double helix, it's got 2 strands. What about the red guy? Is this DNA? Is this RNA? Is this RNA polymerase? Is this something else? Good, this is our RNA. How do we know? Well, it's only 1 strand long. You'll see it's matching up with the letters here. The message is in these letters. So C would go with G, A with, normally it'd be T, but it's going to be U because it's RNA, G with C, and T with A. So let's check one last thing. Which messenger RNA letters would match with this DNA? Select the correct answer. Good, so C matches with G, T with A, T with A, G with C, A with, normally we'd want to write T, but there's no T's in RNA. So it's a U and then T with A. One more. What letters would match with this DNA? So we should have G with C, C with G, A with normally T, but no T's in RNA. So U, T with A, C with G, and A with U. Awesome. That is it for today. Fabulous job.